this is Menchen, and I am here today to present the degradation of mouse locomotor pattern in the absence of proprioceptive feedback as a part of my journal club. The significance of this study resides in the fact that it has been clear that terrestrial locomotion is regulated by both central pattern generator and sensory feedback. However, for locomotor pattern, the influence has not been clear, as of the sensory feedback plays a role in it. Thus, and the purpose of the study is to determine the role of central pattern generator and proprioceptive feedback in controlling the undemanding locomotions like walking and swimming. And this study was not previously conducted because there was no way to separate the influence of central pattern generator and proprioceptive feedbacks. Thus, the research question would be, what is the role of sensory feedback in generation of normal locomotion patterns? Here are some background knowledge to help you understand more of this research. Our independent variable here are proprioception, which are the sensory of body position, movements, and muscle forces. This is sensationed by two receptors in muscle. One is Golgi tendon organ, in charge of sensing tension exerted on the muscle, and another is muscle spindle, which is in charge of sensing the change in length and the rate of change in length for the muscle, which would result in change of our dependent variable, the locomotion patterns. And locomotion patterns are also separated into two divisions. One are the swing stance coordinations of muscles, and the second one are cross-joint coordination, which would decide the position of limb placement. Two of our dependent variables are quantified and measured by EMG and joint angle, recorded from implanted EMG electrodes and video of walking. Here is a brief conclusion to swings and stance, which swing is the phase of walking where your leg is in the air, and stance is the phase where the leg is on the ground and pushing the individual forward. The methodology here used in this literature to separate the influence between central pattern generator and proprioception, and especially between muscle spindle and Golgi tendon organ, is that three groups of mice are created for the purpose of the study. The first group are controlled group, which are the wild type mice. They would have both sensory from muscle spindle and Golgi tendon organ. The second group are the EGR3 mutant mice, in which their group 1A and 2 muscle spindle activation are selectively impaired, so they would still have a intact Golgi tendon organ sensation, but not from the muscle spindle. And the third group are the proprioception killed mice, which all their proprioceptive afferent were eliminated, so they would have no sensation from muscle spindle nor Golgi tendon organ. And the performance of all three groups of mice are compared also between their walking and swimming, which swimming would consider to eliminate the effect of Golgi tendon organ as less force are exerted on the muscles. And EMG data processing are introduced in this slides here. The data collection are conducted through continuous voltage recording from implemented EMG electrodes on the mice, and the three muscles especially focused on collecting were the three flexor muscles, and the first one would be the IP muscle, which moves the hip, and the ST muscle that moves the knee, and the TA muscle that moves the ankle in walking and swimming. Data processing are conducted so that raw data are filtered for noise and amplified, and all values are changed into absolute value for analytical purposes, and burst detection are done to detect the duration and timing of the muscle activation bursts. Here are the results. This page is to discover the effect of muscle spindle, in which we compare the walking activity for wild type and EGR3 mice. The EMG showed us that the activation of ankle flexor has a later offset during the swing phase. As of joint angle, it has been discovered from the video footage that higher lifting feed occurs during walking, and there is also a bigger flexor angle for both hip joint and knee joints, and limb placements has more mistake due to wrong timing of muscle activation. Here we display the results that are related to the Golgi tendon organ. For the wild-type mice, when we compare the walking and swimming activity, the onset of the muscles are changed. As in, for walking, none of the muscles were activated in stance. However, in swimming, all three muscles are activated in sequence throughout the cycle. And for ERG3 mice, all muscles are activated together in the swimming activity. Thus, when we compare the three groups of result, we would discover that the onset of flexor muscles are influenced by both muscle spindle and Golgi tendon organ. Here we display the result for the proprioceptive killed mice, in which all three flexor muscles started to have synchronous onset and offset timing for both swimming and walking. Here we are able to come to some of the discussions from our evidence. When we compare the wild type and EGR3 mice in their walking pattern, we could tell that the only muscle influenced was the offset of the TA muscle. So we would know that the swing to stance transition, which is the offset of flexor muscles, is selectively controlled by muscle spindle. And in swimming, when we compare the EGR3 mice with the wild-type mice, we could come to the conclusion that the stance to swing transition, which are the onset of flexors, are controlled by both muscle spindle and Golgi tendon organ. And the author hypothesized that this is because muscle spindle needs to sensation the change in hip angle, while Golgi tendon organ needs to sense the decreasing of muscle load on the leg muscles. The second group of evidence is that for ST muscle, there is a prolonged muscle activation for proprioceptive killed mice. However, there is no change in ST muscle in the ERG3 mice as compared to the wild-type mice. Thus, we would determine that the knee flexor, which is the ST muscle, is controlled by feedback from the Golgi tendon organ instead of the central pattern generator as considered before. And the last group of evidence would be on the cross-joint coordination, in which the period where TA and IP muscle are both activated is longer when we compare the wild-type mice versus the ERG3 mice. We would come to the conclusion that ankle and hip joint coordination are controlled by muscle spindle, and the period when ST and IP are both activated is longer, so the hip and knee joint coordination are controlled by GTO. Some future research questions that can be asked on this replicable research would be the exact role of central pattern generator and normal locomotion, and how does mechanical load interact with proprioceptive loss? What does imply for human disease, including proprioceptive loss, and how the sensory feedback stabilizes CPG? Here we would evaluate the literature both based on the references. So the thoroughness of the reference is very good. They comprehensively covers for the topic and the methodology, and it is very directly relevant to the research question, and it includes the foundation and current work on the genetic and physiology studies, while the area lacking for the references are the broader modeling and comparative locomotion pattern field, but not necessarily included for the goal of this paper. And here are my references.